And can you see the screen?
Yes, we can.
Okay, so um, in the last lecture, um, I addressed three questions. The first one was, what is a bioreactor? And I showed you the parts in a bioreactor. The main point I wanted to emphasize there is that a bioreactor, unlike a typical shake flask, is equipped with things for controlling the momentum, the heat, and the mass transfer, more precisely, the transfer of oxygen. And then the second point was to explain why you need a bioreactor. And I gave two reasons over there. One is because you need to be able to control these transfer processes for scale up. Not control, but frequently you need to enhance the momentum, mass, or heat transfer and scale up. So anytime you go to a big reactor, big scale, you just have to have these things. And then the last point was, how do you describe these bioreactors mathematically? Starting with the simplest one, I started with a chemostat, and I arrived at the equations, which I will repeat over here. This is our starting point. So we're starting with the equations, which I will simply repeat. Um, this is our system. It's well stirred. It has a stream coming in, a stream going out. Coming in, flow rate is F. The growth limiting substrate is at a single concentration. This is the single growth limiting substrate. It is at concentration of SF, XF, and PF are the concentrations of the biomass and the product, which are assumed to be zero. So it's a sterile feed with no product in it. The product is made exclusively within the bioreactor. So you have cells, and then of course air bubbles. This is the sparger pushing air through it, and what's coming out is at the concentration F, S, X, and P. This is the flow rate coming out, which is equal to the flow rate going in, and then the concentrations inside the reactor, which are the same as the concentrations coming out. So here also we have the concentrations S, X, and P. So we're assuming the reactor to be well mixed. Now, with this, I basically showed last time that if you take mass balances over the control volume shown by the red box, which is basically, this is the balance over, actually strictly speaking, it's a balance over the liquid phase of the reactor, but the volume of the gas phase is so small that you can simply ignore it and assume this is over the total volume. But ultimately, you arrive at the equation. Mass balances we got were dx dt equals d times sf minus s minus mu times x, where mu was defined as the specific growth rate. So mu was defined as the specific growth rate, which is how many grams dry weight do you make per hour per gram dry weight of the cell. So this is like what people call per capita growth. So this is actually a true measure of the fertility. The next thing was, oops, I'm confusing. This is wrong. Yeah, nobody's speaking up. This was minus dx plus
mu x. This is d times SF minus S minus RS times X, where RS, which we call the specific substrate consumption rate, is grams of substrate consumed per hour per dry weight of cells. And finally, this I told you to do on your own. This is grams of product that you make per hour per gram dry weight. So over here, I should also mention where D is defined as the flow rate divided by the volume. So this can be viewed as this is the volume of the reactor. So this can be viewed as reactor. This is actually, let me say, flow rate in reactor volume. How many volumes of the reactor go through per hour? And this is equal to 1 divided by, I don't know what is the notation we use, theta, which in chemical engineering is referred to as mean resonance time. So D and theta are just inversely related to each other. Okay, this is just a review of what we did last time. And I think I also mentioned that I wanted to make a distinction between the hydrodynamics and the reaction. And in this particular case, all the hydrodynamics disappears the moment you say that there is no flow into and out of the system. So there's no transfer. So if there is no hydrodynamics, if there is no advection or transfer, then basically D equals 0 and you get only reaction taking place in your system. So you basically, the moment you put D equals 0, all these terms disappear, the ones which have a D in them, and you're left with dx dt equal to mu x, ds dt equal to minus rs times x, and dp dt equals, something has happened, dp dt equals rp times x. So now this is a problem. Now, so that means in a batch reactor, you basically have a problem. This is a problem of pure kinetics and stoichiometry. There's nothing else going on because you've eliminated all the transfer processes. Okay. Should actually maybe correct myself. There's transfer going on. This is the transfer of gas. We're ignoring it for the time being. I'll deal with that at a later time. So now the question is, how do we proceed further? Okay. So how do we proceed further means you cannot proceed further unless and until you say something about mu, rs, and rp. Only when you specify these can you proceed further. So this is the heart of kinetics. It is saying what are the specific rates at which the reactions, or super reactions, whatever you want to call them, are happening in the system. So now I'll proceed kind of systematically. So the simplest to need specify mu, Rs, and Rp. By specify, I mean you must be able to express them in terms of, the existing variables, x, s, p, so on and so forth. Now, the simplest model first is what one might call a single reaction. This will seem kind of bizarre, but actually it works. Now, this is the simplest one. And this, the essence of it, actually goes back to what Jacques Monod did in 1942. What he said was that the process of growth, which you know from biochemistry to be a very, very complicated set of reactions, he found experimentally that it could be expressed as a kind of a reaction, which is kind of puzzling. So basically, he claimed that S plus, we'll fill up this rest later on, just give you what he found, goes to biomass. And this will also have additional components in it, which we will fill up. What he found was that a given amount of substrate always gave rise to a given amount of biomass. So the conversion of the substrate into biomass seemed to follow a stoichiometric relationship. So you can say that if I define a quantity y, and this is, just notice that I'm using a slightly different notation. When I write yxs, this is a constant denoting how much substrate is consumed in order to produce unit biomass. So this is substrate consumed, I should say grams of substrate consumed per gram dry weight of biomass produced. And what he found was that actually there was a relationship between the two. I mean, if he did an experiment in which he plotted, this is the plot. Here you put down substrate consumed, and here you put down how much cells, how much biomass you produce. You find that the more you give, the more biomass you make, up to a very good degree of approximation. So you can already see that it seems to have the elements of what you might call a reaction. This y-axis appears to behave truly like a stoichiometric coefficient in a reaction, like the reaction 2 times H2 plus O2 gives you 2 times H2O. Except that this y-axis is not an integer. It is just a real number, right? I mean, okay, so the first thing he found was that the ratio shown above is constant. Okay, and the ratio shown above meaning this y-axis. Anytime you consume a certain amount of substrate, you always make a certain amount of biomass. This is what he found. Okay, so we are done with part of the problem. What he also showed was, now this reaction also has a certain rate, right? We have to say what the rate of this reaction is. And basically, let's put a mu over here. The mu is describing the rate at which biomass is being made per unit biomass. The second thing he showed was that the specific growth rate so, and I should emphasize, under the conditions in which only one substrate is limiting. So that means that all other things are in excess. Under these conditions is given by. So basically, this thing has the shape. He basically said this mu, the specific growth rate mu, right? It basically has this shape. It depends only on the concentration of the substrate. So as the substrate concentration increases, this mu increases until it reaches a flat value mu m. And he said that to a first approximation, his data showed that this functional relation could be expressed by a hyperbolic function of this form. This is what he gave the name maximum specific growth rate. And this is what he called the saturation constant. So you can see that this mu m is the specific growth rate that they will reach if the concentration of the growth limiting substrate becomes relatively large. And intuitively, you can think that basically what is happening here is that beyond a certain point, the transporter enzymes that live on the surface of the cell, that actually take the substrate, the growth limiting substrate, and shove it inside the cell, these transport enzymes get totally saturated with the substrate. And because of that, if you add any more substrate to the surroundings, you will not see any improvement in the growth rate. That explains why you get this quantity called mu m. This quantity called Km, which he calls saturation constant, is actually a measure of the affinity. I mean, how avidly does the cell pick up the substrate? So if Km is very, very small, then relatively small values of the substrate will cause the saturation of the growth rate, right? So for example, actually we can make it mathematically more precise. The Km is the value. If I put S equal to Km in this equation, you can see that this becomes mu m multiplied by Km divided by Km plus Km. So that is equal to mu m divided by 2. So Km can be viewed as that substrate concentration at which the specific growth rate is approximately half its maximum value. This Km gives a measure of the affinity. So if the Km is small, it means it will at very low concentrations of the substrate, it will reach saturation. Is that part clear? So now I've taken you to the point where this is the picture we have. Now this picture already allows you to understand part of the problem. Why? Well, we have already specified what mu is. Mu is specified by this equation here. This is call this equation 2. And the second equation is actually right here. I'll call this equation 1. It says that if I know the rate at which X is being produced, then I automatically know the rate at which S is being consumed because they're stoichiometrically connected. So let me write that down in short form very, very briefly. What we have shown, let me just repeat, this is equation 2. We have shown that mu is a function of S, which is given by mu m multiplied by Km plus S. So the growth rate is completely determined by the concentration of the surrounding growth limiting nutrient. But at the same time, we have also specified what RS is. Because what is RS? It is simply Y XS multiplied by mu. This is also a second thing that he found experimentally. So now that means that I have both mu and RS available to me. This is equal to mu XS multiplied by mu m multiplied by S over Km plus S. Okay. Are we good so far? Let me just pause to ask. Is it clear?
Yes, sir.
So far?
Yes, sir. So basically, that specific growth rate follows something like Michaelis-Menten kinetics.
Actually, he even pointed out in his paper that he suspects. Well, he doesn't say he suspects. He says that people might argue that this is essentially a reflection of Michaelis-Menten kinetics, and that this is a reflection of the effect of a single enzyme, right? So basically, one enzyme is rate limiting. So, for example, it might be the enzyme, the transport enzyme that is picking up the substrate. But then he dismisses it, saying that there are so many reactions that it would be dangerous to say that this is due to any one enzyme. So he basically says that empirically this works, and I'm going to proceed. So it's actually quite remarkable that it works. I mean, it's amazing that given the complexity of growth, the fact that you can capture at least any slice of it by assuming growth to be nothing but quote unquote a super reaction, it really is quite, to me, quite remarkable. And, it's worth asking why. I don't think people ask this question a lot, but maybe we can have some discussion of it later on. Any other questions? Okay. So now, all this happens in the 1940s. But actually, subsequently, people argue. So this is now, say, one single reaction. Now, later on, people find that under certain conditions, well, actually, under many conditions, I shouldn't say certain, I should actually say, because this happens, it's really quite amazing that most of the time you will use this model, that under most conditions, the oxygen and CO2, so maybe I should say oxygen consumption and CO2 release rates are also proportional to growth. In other words, stated differently, this equation here can be extended, and I can write it in the form y. This is xs s plus. Now he says that even the consumption of oxygen and actually I should even so rates, and here I should write as well as nitrogen consumption rate
which I'm going to put here. So this is Y xn times, say, ammonia, which is the nitrogen source. All of these, they go to, right? And here I have CO2 plus dot dot dot. Actually, I should even add that people say that even the product, but I will correct myself. This is the least reliable of all of them. So this is the general picture. Now they say that all works out. And now let me maybe point out as to why it works. So what they are basically saying is it is not just the consumption of substrate that is coupled to the production of biomass. Everything else is also coupled to the production of biomass. So basically, if you produce a certain amount of biomass, you will consume a certain amount of oxygen, which is then captured by this stoichiometric coefficient. It's not really a stoichiometric coefficient, but empirically it seems to follow that rule. If you make so much biomass, you will consume so much oxygen. And this coefficient basically describes it. It says how much oxygen you consume per unit biomass. This is the next one is how much nitrogen you consume per unit of biomass. This is how much CO2 you produce per unit of biomass. And this is how much product you make per unit of biomass. So everything apparently, and I should emphasize that this is to a first approximation, works out. Well, why does it work out? It turns out, and this is kind of shocking, at least to someone the first time, depends on. So, why does it work? Why does this simple picture? And so, all this is happening at the rate mu of S if you have one growth limiting substrate. So, actually, I should point out, this is worth noting. See, the everything else in this reaction, as I have written, has well-defined composition, right? This S might be glucose. O2 by well-defined composition might mean it's a well-defined compound. S might be, for example, glucose. O2 is the well-defined entity called oxygen. NH3 is ammonia. Then I have CO2, and then a P would be a well-defined product like maybe ethanol or maybe glycerol or what have you. The only thing that I have not given a composition is this quantity biomass. What is biomass? So now I want to show you that surprisingly, it turns out that the composition of biomass is essentially invariant. It is the same. It seems to have the same relative amounts of carbon, hydrogen, oxygen, nitrogen, so on and so forth, over a wide range of conditions. So let me show this to you straight from the data. So this is showing you the data for the elemental composition of biomass for several microbial species. This is taken from, I think I've given you this reference. It is a book by Viladson, Bioreaction Engineering. This is table 3.2. And I think it has a very good discussion of this stuff. So on the left hand side, he's showing you the column, the different organisms. So Candida utilis is a kind of a yeast. Klebsiella aerogenes is a kind of a bacterium. Saccharomyces is another kind of yeast. E. coli is another kind of bacterium, so on and so forth. The rightmost column is showing you the conditions in which it was grown. And he's saying that the growth was limited by the substrate glucose, and the experiment was performed at a dilution rate of D equal to 0.05. in a chemostat. Um, and this ash content, we can ignore for the time being. We'll come back to it in a little while. But see the elemental composition. The first thing you see is the first four rows show that the elemental composition really does not change much as they change the conditions of growth, whether it is growing at D equal to 0.05 or it is growing at D equal to 0.45. Look at the relative amounts of carbon, hydrogen, oxygen, and nitrogen. Now, you might ask how they measure this. Basically, you take cells, you kind of dry them, and when you dry them, not dry them actually, you heat them to a very high temperature. Then the carbon gets converted to carbon dioxide, the hydrogen gets converted to water, the nitrogen gets converted to, I suppose, nitrogen dioxide. All these things get oxidized. Then you pick them up in a detector. And this ash content is the stuff that does not volatilize. There may be things that never really burn. So that's called ash content. And the elemental composition is what you pick up in this way. I think there is we have one in the instrumentation lab, an elemental analyzer. You basically just take biomass, put it into it, allow it to sort of just burn, and automatically it'll report how much carbon, oxygen, and nitrogen it has. And what is important here is the first four rows show that this elemental composition does not vary much with the conditions up to a point, which I will make clear later on. What is also interesting is that if you compare the numbers for the first strain, which is Candida utilis, and the second strain, which is Klebsiella aerogenes, there's hardly any difference. I mean, the first one is CH 1.83, O 0.46, N 0.19. Now take a look at the first row of Klebsiella aerogenes. It is CH 1.75. It differs only by 0.08. The next one is O 0.43. The other one is 0.46. There's hardly any difference. N 0.22. The other one is N 0.19. So this means, and you can go down all the way. The numbers hardly change. I mean, take a look at Penicillium chrysogenum. This is the strain that is used to make penicillin. Here for H, you have the coefficient 1.64. And for Candida utilis on top, you had 1.83. O is 0.52. In the first one, it is 0.46. N is 0.16. And for the one on top, N is 0.19. It's really not varying a whole lot. Because of this, you can, so this is a purely empirical result. There is no more to it. It doesn't have, anything fundamental associated with it, like chemistry, right? That hydrogen is combining, two hydrogens combined with one hydrogen. It just turns out that biomass has a well-defined composition, purely from experiment. And therefore, people often use what I have shown over here. Right at the bottom, this is the average composition of biomass. People just assume that whatever cell type they have has this composition. See, this
So I just had one question. So why is phosphorus only present in some strains, and why do they not consider it to be a part of biomass?
Oh, well, no, nothing. They just happen to be showing it. They also consider phosphorus.
Okay.
They consider phosphorus, sulfur, magnesium. I mean, all this is available. And that's actually important. The fact that the biomass composition does not change much, that is what allows you to get away with treating growth as basically quote unquote a reaction. The biomass is behaving like a compound with a stoichiometry. Are you following what I'm saying? I mean, maybe we'll get back to it later on. And the second point is, I'm digressing here, but we'll get to this later on. You can see the fact that biomass has a more or less invariant elemental composition
also means that when you want to design, so I'm now getting to the point of, I just told you in the last lecture that people always design the medium, the nutrient medium, in such a way that only one particular substance is limiting. Maybe glucose, or maybe, the nitrogen source, or maybe oxygen. Well, how do they do that? Well, once you know, how much of each element is present in biomass, okay? Then you can easily estimate that if you have so much biomass, you need so much of element, right? I mean, biomass has, let's say, so much phosphorus. Now, if you have some way of estimating what will be the total amount of biomass in your reactor, you know, okay, I have to provide at least this much phosphorus simply to manufacture the biomass, right? Provide the phosphorus that is required to make the biomass. And what you do is you calculate that number and you multiply it by 10. That then particular element is now in a tenfold excess. So you have eliminated it from the picture. And the one element for which you don't do this, that becomes the growth limiting element. So this is like a crude, intuitive idea of how you define media that make one particular element growth limiting. So you might say, for example, I have a carbon limited culture. A carbon limited culture is a culture in which nitrogen and phosphorus and sulfur and everything else is in a tenfold excess. Using these ideas, the tenfold excess means it's 10 times the amount that is required to make biomass. And you can do this calculation only because biomass has a well defined composition. So we'll come back to it later on, but get the basic idea. So coming back now to yeah. So the first reason why it works is that so reason one, the first reason why it works is that biomass has a well defined composition over a wide range of conditions. Now, so I'll the next part, once we go beyond this part, we'll see that this model does break down. It's actually quite miraculous that it even works. And when it breaks down, it is either because A breaks down, which is to say the biomass composition does not remain invariant, it starts to change drastically. And the second reason is that actually, over a wide range of conditions, these stoichiometric, the other stoichiometric coefficients, I should say, uh, uh-huh, the the y's shown above, so behave like real stoichiometric coefficients. Well, that basically means that they're pretty constant over a wide range of conditions. So that means that, so first of all, this X, let me just repeat it, has a very well defined composition of the form CHAOB where I gave you the values of A, B, and C. Let me just give them to you right now. This is 1.81, 0.52, and zero point 0.2. So this is
This is 1.8. This is 0.5, and this is 0.2. So to a first approximation, this holds. And to a first approximation, it is also true that these y-axis, y-xs, y-xo, y-xn, y-xc, and y-xp are constant. This assumption also holds to a very, very good degree of approximation. Well, once you have that, then the model reduces to a really, really trivial, right? All you have to do is a single. So this is what I mean by a single reaction. Growth behaves like a single reaction. Its rate is given by mu of s, right? I've written mu of s actually. I should, it can also be mu of O2. It can also be mu of NH3. It depends on which one you have made your growth limiting substance. So it can be carbon limited. In that case, you would write mu of s. It can be nitrogen limited. Then you would write it as mu of n. But whatever be the case, that arrow on which the reaction rate sits, that rate is basically a hyperbolic function of the growth limiting entity. That tells you the rate at which X is being made. And all other substances are determined by the rate at which X is being made. Because the moment you say, I am making X at this rate, then all the others are determined. SO2, NH3. So maybe I just put down, I'm really belaboring this, this must be obvious. I mean, the principle I'm using here is if I have a reaction of the form 2H2 plus O2 goes to 2 times H2O. The moment I specify, and let's say this is the rate at which the reaction takes place, and let's say this reaction rate depends on, actually you would say that it depends on both H2 and O2, but supposing I maintain O2 in a large excess. So in reality, R depends on the concentrations of H2 and O2, right? This is what you would expect. But supposing I maintain O2 in a large excess, if I maintain O2 in a large excess, then it depends only on H2, right? So then this quantity then that just becomes R of H2. And the moment I specify what, the moment I specify this reaction rate, you have already determined the rates of all the entities. I'm only specifying the rate at which H2 is being consumed, but that immediately determines the rate at which O2 is consumed and the rate at which water is formed. Is that part clear? I think I'm kind of really belaboring this. So, is that part clear? If it is, then we can move to the next step. So you got
Yes,
the
that's
basic
clear.
idea? Okay. So now let's use this to actually see what happens in a shake flask. Once again, I think you have not done the lab, so maybe you may or may not be able to relate to it. So let's see. So now use the above model to understand what happens when you grow cells, when you grow cells in a shake flask. Now, a shake flask is basically a batch culture. They do a pretty good job of mixing. So the constant you can assume to a first approximation that what you have is constant concentration inside the flask. Okay, so now here's how you typically do the experiment. You typically take a flask,
You put some medium into it. This is the nutrient medium. And now in a shake flask, actually everything is in excess. Meaning, I'll make that clear in a minute. I'll make this more precise in a minute. Um, so the nutrient medium contains, a source of carbon, a source of nitrogen, a source of sulfur, a source of phosphorus. All these things are there, available through some compound. And then you autoclave it, you sterilize it, so you kill all the organisms that might possibly be inhabiting the medium. So then you autoclave, and then after you autoclave, you basically get a sterile medium, and then after you autoclave it, then you inoculate it. So inoculate it means you add a small quantity of cells, really small, and watch them grow. So this is the experiment. Now the important point here is that in the nutrient medium, everything, all the elements are in excess. What do I mean by they are in excess? So in particular, so supposing you had, going back to this equation, maybe this is the right way to put it. Supposing you had this type of an equation for every element, you know, basically for every element, if the concentration is sufficiently small, the growth rate will depend on it. But as you make it larger and larger, it will become independent of it. What we are basically saying is that the concentrations of all the compounds are so large that none of them are in this Km region. They are all well beyond the Km region. So essentially, the cell will grow at its maximum growth rate, mu m. Is that part clear? Did I make sense?
Yes, sir.
Actually, I'd be happy to hear something from other people too. Dave, are you the one who's the? I can't see actually.
Yes, sir.
Can you follow what's going on?
Yes, sir.
And I see. Bhavya, are you able to follow what's going on?
Yes, sir.
Okay. So now, coming back to what I was talking about. Okay. So now when you do this experiment, here is what you find. So I'll show you what you observe. Suppose, were these experiments done online, or will you do these experiments when you come back?
No, so they'll be done when we come back in the
All
winter.
right, so you'll see this. So you inoculate it at time t equal to 0 with some very, very small quantity of cells. So what you find is that basically, let's say this is your cells, which are typically measured, cell densities are typically measured using a spectrophotometer, which basically is nothing but light scattering. So you have cells sitting in a cuvette, light shines on it, it comes in at I0, goes at I, and it follows the Beer-Lambert law. So basically, you measure, essentially, the spectrophotometer spits out a quantity called absorbance, which is proportional to the log of I0 over I. And that tells you a little bit about how many cells you have, right? Because this is the fold change, log to the base 10. So if you measure log of I0 over I, you're basically measuring the fold change in the light intensity as it passes through the cells, right? So if this quantity I0 over I is equal, this quantity, if this quantity is equal to 2, it means you're getting 100-fold change in the light intensity. That means I0 over I has gone down by a factor of 100. And that tells you a little bit about the density of the cells. So this is actually measured using what is called OD. This is also called OD, or optical density. Optical density. And they frequently put a little number below it. They may say OD 600. That means that 600 represents the wavelength of the light that is being used. So this 600 here is light wavelength. So you can use 600, you can use 500. Hopefully, this will be covered in your practicals. Okay, so now when you do the experiment, and you take, so you start off with, let's say, some x0, and then so I should point out that, by the way, this OD 600 we know is directly proportional to the grams dry weight per liter. Actually, in the lab, we measure this quantity, but we know this to be true. This is done by creating a calibration curve. So as you do the experiment, you measure the OD, and then from the calibration curve, which you have constructed earlier on, where you took samples of various OD and measured their dry weights, from this calibration curve, knowing the OD, you can back out the concentration in grams dry weight per liter. So this graph kind of looks like this. And it goes up, and this going up, actually, it turns out, is actually exponential. Okay? And then it's exponential literally till the end. And then it craps out. It's actually easier to see this if you plot it on a semi-log plot. You look at it on a semi-log plot, then basically this graph looks like there's almost a discontinuous change, like it goes, you can barely observe, basically it kind of goes like that. So that means this is the, you can say that this is the part that is exponential, and this is the part that they give the name stationary. And if you wait still longer than that, then this starts to decline. That's the death phase. So roughly speaking, what is happening is this is actually not unlike what you might expect in a human population that is subjected to a fixed quantity of food. You first grow on it until it is exhausted. Once it is exhausted, you may be able to survive for a while by making use of the reserves within your body. But once that phase also goes away, which is the stationary phase, then people start to die, and this is called the death phase. So I should point out that actually, even though the subject of microbiology has been around for, I would say at least 150 years, if you count Pasteur, 90, so 90, I would say 99% of the studies since the 99% of the studies have been
on the exponential growth phase. The rest of the stuff, the stationary phase, the death phase, actually there can even be a little lag at the beginning. Nobody studies that stuff. People have ignored it for years. I suppose it's because the most interesting thing these bugs do is they grow. So um, maybe that's the reason why. So we don't know a whole lot about what happens here and what happens here. What I'm telling you is just superficial stuff. In a deep, fundamental way, we know much more about the phase of exponential growth than we know about anything else. Okay, so now can we explain this data and we explain these data? Actually, maybe I should actually give you some more before I proceed. Now, suppose in addition to measuring just the biomass concentration, suppose you also measure, and I will erase this. Suppose you also measure the concentration of, say, glucose. Okay. So I'm going to show it over here. So now the first thing I want to point out is that during exponential growth, as I pointed out to you, everything is in excess, right? There is no shortage. All the compounds are in excess with respect to their saturation constant. So the rates at which they are taken up are at maximum levels. And therefore, what you see is the maximum growth rate. However, this particular stationary phase, which is reached, the stationary phase, which is reached over here and over here, the stationary phase starts because one of the substances has finished. It could be the carbon source, it could be the ammonia, it could be something gets over. When that something gets over, growth will stop because it needs all the elements. It cannot grow if 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 nitrogen is not there, it can't make proteins. If phosphorus is not there, it can't make DNA. Do you get what I'm saying? So the exhaustion that takes place due to one of the nutrients. So this is a different kind of limitation. This exhaustion that is taking place here is because of which of the substances ends first. If that substance, so whatever that substance might be, let's say if you also plot so this is, it's a little bit, so this is, so this is the biomass, and this is that particular substrate, which again generically I'm denoting as S, the exhaustion of which leads to the onset of the stationary phase. So this is a limitation in another way. So one is the limitation on the rate of growth. The other is the limitation on the exhaustion of growth. I hope you can see the difference. So frequently we design the medium in such a way that this experiment, the stationary phase kicks in when the carbon source is exhausted. That would be typically S is the carbon source. But it need not be. You could design the experiment such that something else is exhausted before glucose. If that happens, well, glucose will be left over when the experiment ends, right? If I design the experiment in such a way that the stationary phase occurs when uh, nitrogen is exhausted, you will find that at that point in time,
There's a whole bunch of glucose floating around in the medium. So we have these two curves, and we would like to see how our model allows us to understand what is going on. So I'm going to go back to these equations now. Start from the beginning. So I'm going to work with uh, this equation and this equation. I'm not going to work with the equation for P because there is no product. And what else am I going to work with? Well, I can't do much with these equations unless I know what mu and RS are. But I have already given that to you. I told you that mu m is given here and RS is given here. So I have, let me repeat, dx dt equal to mu x, ds dt equal to minus rs times x. Two equations, differential equations. But they leave me with nothing unless I say what's happening to mu and rs. But mu and rs also, we have postulated, have a form. Mu s is mu m times s over km plus s, and rs is nothing but a multiple of mu. So we have everything we need. Well, how do we proceed? So we have dx dt equal to mu times x, where mu we said was equal to mu m times s divided by ks plus s. Actually, I should correct myself. I think I wrote km. Uh, they never use in microbiology. They never use km. They always use the notation um, ks for saturation constant. So this should be ks, and this should also be ks, and this should also be ks. Okay, so ks for saturation constant, and we also have ds dt equals minus rs times x, where rs is nothing but y xs multiplied by mu. These are the two relationships. And my initial conditions, time t equal to 0, I have the relation x at 0 equals x0 and s at 0 equals s0. So going back to the graph, you can see what x0 is right here. And let me also show you clearly. This is S0, initial concentration of the substrate. Okay. Now notice the way I have drawn it. The way I've drawn it, x0 is really, really small, and s0 is very, very large. Actually, I can put numbers to it. Normally, the experiment is done with s0 uh, on the order of 1 to 10 grams per liter. And x0 is very, very small. It is on the order of something like 0.005 grams dry weight per liter. That's how we typically do the experiment. Now, if that is true, then here is what you need to see. At time t equal to 0, take a look at this expression. At time t equal to 0, oh, I should mention something else. So S naught, I should put down here, is on the order of 1 to 10 grams per liter. I should also give you an estimate of this parameter value, Ks. What is it typically in nature? Well, this quantity is extremely small. This is on the order of 10 micrograms per liter for bacteria. Maybe I should say 10 to, this is 10
to 100 micrograms per liter for bacteria and 10 milligrams per liter for yeasts. Stated differently, what I'm saying is that these bacteria can basically snap up, they can sweep up everything they have in the environment when the concentrations are really, really small. They have extremely high affinities. So if I start the experiment with like 1 gram per liter, which is orders of magnitude bigger than Ks, then it is clear from that expression over here that when you start the experiment, mu will be equal to mu m, right? Because that S is so much bigger than Ks that that ratio is nearly equal to 1. Are you with me so far?
Yes, sir.
Okay, so now ask yourself the question, how long will this system sit? So this calculation I just did was with S0, which is the concentration at time t equal to 0. As time proceeds, obviously the concentration of the substrate will go down, right? So let's say I start with 1 gram per liter. After some time, it will become 0.9. Yeah. After some more time, it will become 0.8 and then 0.7. Do you think that this ratio S over Ks plus S is going to depart significantly from 1 if the concentration goes down from 1 to 0.9?
So from 1 to 0.9, it won't depart as significantly.
Will it depart significantly from 0.9 to 0.8?
Not really, sir.
So that means that this situation is going to prevail. Will it change significantly when S becomes equal to 0.1? Keep in mind that 0.1 grams per liter is 100 milligrams per liter. So that means even when the concentration has gone down to 0.1 gram per liter, it is still much larger than Ks. So that means that basically mu will remain equal to mu m until S goes down to a number comparable to Ks. After all, as long as Ks is much bigger than Ks, the ratio is definitely equal to 1. So mu is essentially equal to mu m and does not depart from it unless S goes down to a value comparable to Ks. But Ks is so small that that is literally at the end when all the substrate has been exhausted. So that means that to a first approximation, I can actually find out what is happening literally all the way to the end simply by putting mu equal to mu m. And that really simplifies life. So basically, since S naught is typically much, much bigger than Ks, growth is exponential. I should actually say is exponential, i.e. mu is a constant. Mu is approximately equal to mu m, which is a constant value. So mu is essentially constant, so it grows with constant exponent, right? Until S becomes comparable to Ks, which is literally the end of growth, which is essentially uh, the end of growth, the end of the growth phase. Let me repeat by going back to the picture. So because of what I'm saying, that is why you see that it basically, the line, maybe geometrically what I'm saying is that the observed behavior is the way I have shown it, which means you have basically a straight line and then a horizontal line. You do not have, and let me draw that in red,
you do not have this picture. Ah. If I had this picture, it would depart from exponential growth, right? This entire area, which I have put in with the vertical lines, the growth is not exponential, right? Because it's just changing from exponential growth on a semi-log plot. That doesn't happen. It basically is exponential all the way to the end, literally, and almost no time is wasted in switching from exponential to stationary. So, stated another way, the transition from exponential to stationary is extremely abrupt. It takes place within literally minutes. This experiment has a time scale of hours. We'll see more of that in a minute. So this experiment will typically last 8 to 9 hours, and the time scale over which it switches from exponential to stationary may be 4 minutes. You may even miss it. You may not even see it if you're doing the experiment. Because you take samples every 1 hour or so. Okay, so now this really simplifies life. That means that basically, to a first approximation, you can describe everything by the even simpler system. So if you only want to know what is happening during the exponential phase, then everything reduces to the form dx dt approximately equal to just mu m times x, because mu is approximately equal to mu m until literally the end, until s has reached a value comparable to, and the second one, ds dt equals minus y times xs multiplied by mu m times x. That's all there is to it. This is what describes the exponential growth phase. And of course, we have the initial conditions x equals x0 and s equals s0. Now, this is really, really easy to solve, right? This tells me right away that x at any time is nothing but integrating the first equation is basically x0 multiplied by e to the power mu mt. Okay, and now I can plug this equation into the second equation, right? So then I have the equation ds dt equals minus y xs multiplied by mu m x of t. So one way to solve it is now plug in x, integrate, and that will tell you how s changes. Can you see what I'm saying? Yeah. But an easier way, right? You have to integrate this with s at 0 equals s0. This is one way to do it. You integrate it out, try it out. But it's much easier to see that from these two equations, it immediately follows that dx, ds, right, is equal to, just dividing it, you get minus 1 over y xs, right? Just dividing the two equations. Now, this quantity notation is such, this is called 1 over y sx, is often called y sx. So just see what I mean by it. Y xs is defined as the amount of substrate consumed per unit biomass. If I reverse the indices, then the way I read it is how much biomass is produced per gram of substrate. And the two will be the reciprocal of each other. It's just taking 1 over it. So this notation makes perfect sense. And this implies, if I integrate it, I can basically write it as dx is equal to minus y sx multiplied by ds. But now I integrate this, right? This is dx, which goes from x0 to, let's say, any x at any time. And on the right hand side, I take it from S0 to any S for the instantaneous S at that time. And you find that basically X at any time minus X0 equals, this becomes minus YSX multiplied by S minus S0. But I'll remove this, put a minus here and a plus here. And you basically get, you can rewrite this equation as y sx multiplied by s 0 minus s of t. Now this equation makes perfect sense. What this equation is saying is, the left hand side denotes the amount of the increment of the biomass concentration. That increment of the biomass concentration is equal to the decline of the substrate concentration multiplied by the stoichiometric coefficient. Well, that implies that x of t equals x0 plus ysx multiplied by s0 minus s of t. Now, this equation can be used to solve for s of t. We already know x of t, solve for s of t, right? Do you follow what I'm saying? Am I clear, or is this hard? I mean,
Yes, sir.
If this is equal to x0 e to the power mu mt, you can immediately solve for what s of t will look like. So I initially told you that you could do it by doing this integration. That is another way. You can check it out, right? Just put in x of t equal to x0 e to the power mu mt. But the point is that in that integration, you do not see that ultimately x and s are related stoichiometrically. That thing is coming through much clearly in this method when you get this relation. It shows you that basically x changes, the change in x is intimately connected to the change in s. And if that is the case, then I can find out the change in s immediately if I know the change in x. But I know the change in x because I have already solved for the differential equation for x. So therefore I know the change in s. Just solve this equation and you're done. You get s also as a function of t. So roughly speaking, what you're finding out is that this, going back to, I said, this is what you observe. Can we understand it? Well, we do. This x increases. So we are only trying to predict what is happening in the exponential regime, drawn now with a vertical red line. I'm not trying to go beyond it. It will crap out. It will not tell you, it won't explain what is happening. It is not supposed to be used beyond that. In that regime, we have just shown that x increases exponentially and s declines exponentially. So if x is going up faster and faster and faster, then that means s is coming down faster and faster and faster. And you can see that in the way I've drawn it. The slope of x is constantly increasing, at least in the exponential regime. The slope of s is constantly decreasing. It's becoming bigger and bigger in magnitude, but its sign is negative. Okay, so that's clear. We'll pick up on it next time. Any questions?
No, sir.
Okay, all right. So we'll meet on Thursday. I believe there is a lecture on Thursday.
Yes, sir. Yes, sir.
Okay, thank you.
Thank you, sir. No, thank you, sir.